Today we're talking about bioenergetics. We'll talk about what that is in a minute. We are recording this session, and the recorded version of this will be posted on our A+ College Ready YouTube channel here within the next couple of hours. If you haven't already muted your microphone, please go ahead and do that and turn off your camera. That just makes the online system work better. If you have a question, though, unmute real quick, ask the question. We want you to ask questions. The presentation I'm using today, I'll send out to teachers here in a minute. So if you would like a copy, just request a copy from your teacher. Our next one of these webinars is going to be a week from today. So that's May the 5th. It'll also be at 2. And you can see the join code there at down there at the bottom. What we'll do on that day is we'll finish up bioenergetics. And then after that, I think we'll have one more of these sessions, and that'll be sort of a general test preparation kind of session. We're going to learn a lot tonight at another webinar about how the test is going to work on exam day. So I'll incorporate that into that last webinar. All right, let's get started for today. So bioenergetics is the field we're talking about. So in bioenergetics, we're looking at how energy flows through living systems. So we're talking about things like enzymes, and that's mostly what we're going to focus on today. Metabolic pathways. We'll talk about what a metabolic pathway is here in a minute. We'll also be talking about cell respiration and photosynthesis. I'm not sure how much of those we'll get to today, but if we don't get to them today, we'll get to them in the next part of this session. But essentially, bioenergetics is about how living things get their energy, how they acquire it, how they transfer it from organism to organism, and how they transform that energy in order to do biological work. Things like move and fly and break down food. Now, as it says here, important note, matter and energy move through an ecosystem together oftentimes. They're in the same molecules. They're in the same organic compounds. They're in food. But they follow different paths. Producers, things like plants and algae, and some bacteria, cyanobacteria, for example, they get matter from carbon dioxide, from water, and from minerals, usually from the soil or sometimes from the water. And their energy comes from light, from sunlight. Heterotrophs get both their matter and their energy from food. We get both there. All of the matter, or at least most of it, is eventually converted back into carbon dioxide, some into water, some into minerals, and some just into waste, metabolic waste, feces, urine, those kinds of things. But all the energy eventually leaves the ecosystem as heat. Now, a lot of people talk about energy getting used up. Energy doesn't get used up. One of the most important ideas in science is the law of conservation of energy. And that law says that that energy can't be created or destroyed, but it can be transferred from one thing to another. And it can also be transformed from one kind of energy to another kind of energy. But the amount of energy that goes into an ecosystem is eventually the same amount comes out. It just comes out in a different form. It comes out as heat, which is really hard to reuse again. So some important things. Matter cycles continuously through an ecosystem. When we talk about matter, we're talking about the atoms and molecules that make up life. The carbons, the nitrogens, the hydrogens, the oxygens. Those things are used over and over again. They don't get used up. They cycle. But energy comes in from the sun, moves through a food chain or an ecosystem, and eventually leaves and goes out as heat, eventually ending up back into space. It's not used up. It's not created. It's transformed from light to chemical energy, probably to a different kind of chemical energy, ultimately to heat or thermal energy, and then it's transferred through the food chain and ultimately out back into space. So a really important idea is this you see in bold down here at the bottom. The complex organization of life requires a constant input of energy. Energy has to be coming in from the outside all the time, And also living things have to exchange macromolecules with other parts of the food chain or the ecosystem. So really important idea, bioenergetics. Here we can see some of that going on. So we see energy coming in. Energy is moving through the food chain from the producers, where the producers lose a lot of that energy as metabolic heat. But some of that energy is transferred to the next level of the food chain, the consumers. During each consumer level of the chain, more and more energy is transferred out as heat. And we can see more of that here. Also, the decomposers break down dead bodies and transfer more of that energy out as heat, until ultimately all the energy that came in comes out of the system in one place or another as heat. But all those nutrients, all those molecules, all those compounds, they are cycling. They're being used over and over again by different organisms and not used up. All right, the main focus of today is going to be on enzymes, and we have an enzyme FRQ we'll talk about in a minute. But first of all, just some basic information about enzymes. First of all, know that enzymes are made out of protein, which means they're coded for by your genes. A lot of genetic disorders are where you have a mutation in a gene coding for an enzyme. And because that enzyme doesn't function correctly, there's all kinds of problems in the body. Enzymes are what we call biological catalysts. They catalyze chemical reactions in your body. They help them to get started, and sometimes they speed them up. They increase the rate of biological reactions. Without enzymes, most of the chemical reactions that make you go wouldn't work. And everything you do as a living organism in one way or another is a chemical reaction. Whether it's a memory, a thought, a movement, an action, all those things are chemical reactions, and all of them require enzymes to help make those things happen. Enzymes work by lowering what we call activation energy, the amount of energy that it takes to get that reaction started. With enzymes, it requires a lot less energy for the reaction to take place than without. Typically, enzymes act upon some kind of molecule, and the enzyme that they act upon is called the substance they act upon rather is called the substrate. Now, if it's an enzyme that breaks things down, if it's a catabolic enzyme, it's probably just going to act on one substrate and break it down into multiple pieces. On the other hand, if it's an anabolic enzyme, one that builds more complex molecules, it may have a couple of different small substrates that fit into the enzyme, and then that enzyme hooks those together by some kind of bonding arrangement. Either way, whether we're talking about one substrate or multiple ones, those substrates bind to a place on the enzyme, a groove on the enzyme called the active site. That active site has a really specific shape, and it also has a really, really specific charge. And the shape of the substrate and the shape of the enzyme have to fit together. They have to be complementary. A lot of times people use the analogy of the substrate being like a key and the active site being like a lock. They have to fit. And this is one of the reasons that enzymes are really, really specific. Each enzyme can only catalyze one specific reaction, because the only substrates that will fit into it are the specific one that it's made to work upon or act upon. So again, this complementarity of shape and charge causes enzymes to be specific, and it causes them to only catalyze one particular reaction, make that one reaction happen. That means in your body, you have many, many, many different kinds of enzymes because you do multiple kinds of reactions in your body. Another important point is once the substrate binds to the enzyme will undergo a very slight change in shape. Sometimes this is called a conformational change. Lately in AP Bio, they like to call it an induced fit. And this helps the enzyme to function. So when this enzyme changes shape, this is when it either pulls apart the pieces of the substrate and breaks it down, or maybe it pushes together two substrates and helps them to bind together. But this happens after the substrate fits into the enzyme. It's just a slight change in shape that helps to catalyze the reactions. I've already mentioned earlier that some enzymes catalyze anabolic reactions. They help to build macromolecules, whereas other enzymes catalyze catabolic reactions. These are breakdown reactions. Now, important thing, enzymes are catalysts. That means they help to get reactions started, but aren't actually participants in the reaction. That means they're not changed by the reactions they catalyze. They don't get used up or broken down. So an enzyme is recyclable. It will be reused over and over and over and over again to catalyze the same reaction. So enzymes don't get used up by reactions. That's an important idea. Each enzyme works really best at what we call an optimum temperature and optimum pH. For enzymes in the human body, that optimum temperature is around 98.6 Fahrenheit or 37 degrees Celsius. The pH is around 7-ish for most of enzymes. Obviously, there are exceptions, like enzymes in your stomach. Their optimum pH is going to be closer to 2 because it's really acidic in your stomach. If the temperature or the pH is moved away from those optimal conditions, the enzyme stops functioning as well. And ultimately, it will start to denature. Denature means it changes its shape and it loses its function. With enzymes, it's really important to remember that structure determines function. Sometimes people say form determines function. If the shape, if the structure of that enzyme has changed at all, it won't work. It won't catalyze the reaction. So denaturation is when the shape changes due to changes in the environment. And here we're talking about major shape changes, not little minor movements of the enzyme. Typically, denaturation is permanent. So once an enzyme denatures, it's not ever going to work again. The reaction is not reversible. There's some exceptions to that rule, but typically it's not reversible. Here's some diagrams just to kind of show us how enzymes might work. So the purple is the enzyme, and you can see the grooves that are labeled active site. Here's the substrate. So this substrate is going to fit into those grooves like a key goes into a lock. Once it fits in there, this enzyme is going to go through a slight change. And what's probably going to happen here is it's probably going to start bending a little bit outward. And what that's going to do is that's going to stress this bind between these two pieces. And ultimately, it's going to catalyze the breaking of that bind to release glucose and fructose. So this is an example of a catabolic enzyme. It's catalyzing reactions that break down, in this case, fructose, break down the fructose, a disaccharide, into its two monosaccharide components, glucose and fructose. Those are the, and I said that right, this is sucrose, which is getting broke down. So this is sucrose. Sucrose is broken down into glucose and fructose. These are the products. Sucrose is the initial reactant. Also notice that this enzyme isn't changed at all from when it started. And that enzyme can be used to catalyze that same reaction over and over and over. The diagram on the bottom is just reminding you that once the substrate and the enzyme bind together, that the enzyme will go through a minor shape change, which helps to catalyze the reaction. Probably going to kind of pull out this way, breaking the bond between those two pieces. That minor shape change, which helps to catalyze the reactions, is called an induced fit. So we already talked about a few factors that affect enzyme function. Let's go through a list though and talk about a few more. Concentration is a big influence. Concentration of both the enzyme and the substrate. Number one, when the concentration of enzyme goes up, the rate is also going to go up. That's true unless you just run out of substrate. But typically, higher concentration of enzyme, faster rate of reaction. Lower concentration of enzyme, lower rate of reaction. Same goes for substrate. Typically, higher concentration of substrate is going to mean higher rate. That's true until you get to the point where all of your enzymes are continuously occupied by a substrate. At that point, more substrate's not going to change the rate much. The reverse logic applies too. Less substrate, lower rate, slower rate. So concentration is a big deal when we talk about the rates of reactions that are catalyzed by enzymes. pH, also a big deal. Remember that enzymes work best at optimum pHs. So if you move that pH up and you make it more basic, or if you move that pH down and make it more acidic, either way, the rate decreases. So whenever the pH is moved away from optimal conditions, you get a rate decrease. The same goes for salinity and for temperature. Enzymes have an optimal salinity. And if you move that salinity, and that saltiness, by the way, in either direction, higher or lower, away from the optimal conditions, the rate's going to decrease. Now with temperature, let's be a little bit more specific about temperature. Typically, when you increase the temperature of an environment, reaction rate goes up. That's true for reactions catalyzed by enzymes. But at the optimum temperature, which for most enzymes in our body is body temperature, 37 degrees Celsius, which we see down here, our enzymes are working their best. They're working their fastest. We get the highest rate of reaction. If you pass that and get much hotter than 37, the enzymes start to denature. So the rate drops dramatically. If you cool it off a bit, the cooling off doesn't really affect the enzyme so much. It slows down the motion of molecules. Reactions don't happen unless molecules collide. Enzymes collide with substrates. So if they're moving slower, reaction rates are going to be slower. So lower temperatures, lower rate. Higher temperatures up to the optimum temperature are going to increase the rate of an enzyme catalyzed reaction. But if you go higher than that optimum temperature, again, enzyme starts to break down, they start to denature, and the rate will drop very dramatically. Here we can see optimum pH as well. For most of our body's enzymes, that's around 7. And if you go either way, if you go lower and more acidic, or if you go higher and more basic, the rate drops very, very quickly. Let's also talk about inhibitors. So inhibit means to stop. Inhibitors are substances that attach to enzymes and essentially stop them from functioning. There's two main categories we talk about. One of them are called competitive. These are substances that are shaped and or charged like the substrate. They actually can fit into the active site. Well, if they're sitting in the active site, that means the substrate can't. So they're competing with the substrate for the active site. And effectively, they're blocking the active site so that the enzyme can't work. It can't catalyze the reaction. Non-competitive inhibitors, on the other hand, bind somewhere else on the enzyme. There are places for what are called cofactors and coenzymes to bind. There are feedback or allosteric sites. Non-competitive inhibitors can bind to one of those. The net effect, though, is when a non-competitive inhibitor is bound anywhere on that enzyme, the enzyme changes shape. And because it changes shape, it doesn't work anymore. So regardless of what kind of inhibitor we're talking about, inhibitors bind to enzymes and essentially stop them from working. I already mentioned cofactors and coenzymes. So a cofactor is usually a mineral, oftentimes like a metal ion, like iron or magnesium or something like that. They attach to specific points on an enzyme and essentially help to activate them. So instead of inhibitors, cofactors and coenzymes are activators. Cofactors are usually minerals. Coenzymes are usually organic. Oftentimes they're vitamins. This is one of the reasons we need our vitamins. They attach to specific spots on very specific enzymes and help to activate them. That's why oftentimes vitamin deficiencies cause all kinds of mass chaos in their body, because certain enzymes that are crucial can't work without those coenzymes, which are vitamins. So again, we oftentimes call cofactors and coenzymes activators because without them attached to the enzyme really doesn't work. Here we can see a little bit of inhibition going on. So here we have a normal substrate, a normal active site. They're going to bind. They'll go through an induced fit, and the reaction will be catalyzed. That's normal. Here we see a competitive inhibitor. So it binds to part of the active site. And because it's sitting there, the normal substrate can't bind. So the reaction is inhibited, stopped, prevented. Here we have a non-competitive inhibitor. Notice it's not bound to the active site. Here's the active site. It's bound somewhere else. But notice that it changed the shape of this active site. And because the shape is different, the substrate won't fit into that groove. And because it won't fit in, this reaction is also inhibited, blocked, prevented. All right, that's a quick run through of enzymes. A couple more points we want to talk about that are related. There seem to always be questions on the AP exam about metabolic pathways. And students get those mixed up. They're confused about what a metabolic pathway is. So first of all, remember that everything that happens in your body or in a cell is chemistry. It's a chemical reaction. Most of these reactions are really complicated, and they're multi-step processes. So a metabolic pathway is a set of enzyme-regulated steps, enzyme-regulated biochemical reactions that lead to either anabolic pathways, build-up, biosynthesis, or breakdown, catabolic pathways. There's lots of different metabolic pathways in your body. You could think of glycolysis as a metabolic pathway. It's part of cell respiration. Krebs cycle, the second part of aerobic respiration. The Calvin cycle, the second part of photosynthesis. Or you could think of the whole process of photosynthesis, the whole process of cell respiration, the whole process of digestion. You could think of those as an overall process as metabolic pathways. But essentially, again, a metabolic pathway is a set of regulated chemical reaction steps that take in a reactant and ultimately transform it to a product. Each step of that pathway is a separate reaction. And therefore, each step requires a different specific enzyme to catalyze that step. So a multi-step metabolic pathway is going to require multiple specific enzymes to make it work. Another important point that shows up on pretty much every AP exam is that many of the major metabolic pathways in all of life, like glycolysis, Krebs cycle, are conserved amongst living things. Glycolysis works essentially the same in all living things, from bacteria to fungus to protist to plant to animal. Same can be said for Krebs cycle. So this indicates that organisms evolved from a common ancestor, that they're related through evolution. The last thing I'll talk about before we get to the FRQs is feedback inhibition. So let's just read the definition that I have here first of all. Notice also, though, that sometimes you'll hear this process called allosteric inhibition. It's the temporary deactivation of an enzyme or a metabolic pathway brought about by the elevation of the end product of the pathway. We could think of this as a type of reversible non-competitive inhibition. And again, we might think of it as allosteric inhibition. Let's look at when this might be important. So let's say that this end product is something you need in your body, and that each one of these arrows represents a chemical reaction involved in the metabolic pathway that makes substance F, whatever substance F may be. Each of these E's is an enzyme. So this enzyme makes reaction 1 get started. This enzyme makes reaction 2 get started. This one makes reaction 3 get started, and so on. Substance B here is the product of this first reaction. Substance C is the product of the second reaction. D, the product of the third reaction, and so on. We're starting with an initial reactant of A. So what this pathway is trying to do is essentially convert A into F, because F is something your body needs. But here's the deal. Your body doesn't need an unlimited supply of F. Eventually, you're going to make enough. And it doesn't make sense to continue to use resources, to use energy, to make something you already have a lot of. So as the concentration of F increases, it becomes more and more likely that some of F will bump into and bind to this groove. This groove is sometimes called the feedback site or the allosteric site. It's separate than the active site, which we see on the other side. So this allosteric or feedback site is a place where the end product of the metabolic pathway will bind. Typically, it's found, the allosteric site is found on the first or one of the very early enzymes in the pathway. When that end product binds into that allosteric site, it changes the shape of the active site. Well, that means that enzyme 1 doesn't work anymore. Well, if enzyme 1 doesn't work anymore, that means substance B isn't being produced. Well, if substance B isn't produced, there's no substrate for enzyme 2, so it doesn't work, which means that enzyme 3 doesn't work, which means that enzyme 4 and 5 don't work. So by the end product binding to that allosteric site, it essentially turns off the whole metabolic pathway. This is temporary. When the concentration of product F begins to get low, eventually the piece of F that's sitting right here gets used up. And when it gets used up, the active site goes back to its normal shape. Enzyme 1 starts working again. Substance B starts getting produced again. That allows enzyme 2 to have a substrate and work. Well, that produces C. Well, C is the substrate for enzyme 3, so it starts to work, produces D, and we keep going until eventually more F starts getting made to reduce the F that's been used up during the process. So this feedback inhibition is another form of a negative feedback loop. It's essentially a way to conserve resources in the body. Make a material when you need it. When you have enough, cut off production until you need more. It's a negative feedback loop. All right, so now we're going to look at an FRQ. I'm going to blow this up a bit so you can see it. And what I'd like for you to do is to take a minute or two and just read through the whole question, and then we'll take a minute and go through it. All right, so we have this enzyme, alpha amylase, which is found in saliva. And the substrate for alpha amylase is starch. So this enzyme is going to start the breakdown of starch even while it's still in your mouth. It converts starch into the disaccharide maltose. And we can see that at time 0, there's no maltose. So this is right when we put the starch into the mouth. And then after 10 minutes, there's 5.1 micromolarity. That's the concentration. After 20 minutes, 8.6 micromolarity, and so on, all the way through 60 minutes. So the first thing we're asked to do is to graph the data and to calculate the rate of the reaction for the period of 0 to 30 minutes. Now remember, on this year's exam, you're not going to be asked to graph. My guess is, though, you're going to be asked to interpret a graph. So we're going to go ahead and do the graph. I've got an axis on the next slide. I have my numbers written down. So remember, we're looking at time in minutes and maltose concentration in micromolarity. So here's my axis. And remember, on the x-axis, you want to plot your independent variable, which in this case is time. And we're measuring time in minutes. Include your units. Our dependent variable, we want to put on our y-axis, and that is maltose concentration. And we're measuring that in micromolarity. All right, and then our numbers, we had 0, 0, we had 10 and 5.1, and so on. So let's see, I've got 10, 20, 30. I'm going to make each one of these little lines be 5 down here on the bottom. So it's going to be like 5, 10, 15, 20. And I'm going to run out of space. So what I'm going to do is just label the big ones. So if this is 5, 10, 15, 20, this is going to be 25, 30, 35, 40, 45, 50. So this is going to be 75, and this is going to be 100. I think that'll work out okay. And then my concentrations are from 0 to 11.6. So I'm just going to make each one of my Y units be 1. So that means this is going to be 1, 2, 3, 4. 1, 2, 3. This is going to be 5, 10, 15. So now I'm going to plot my points. So my first point was 0, 0. My second point was 10, 5.1. So I'm going to go 5, 10, and 5.1. That's going to be about right there. And then I've got 20 and 8.6. So somewhere right in there is 20 and 8.6. All right, then I've got 30 and 10.4. So 30 and 10.4 is going to be somewhere about right there. Somehow I lost a dot. So I lost 20 and 8.6. Let's put that back. All right, so next I have 40 and 11.1. And then I have 50 and 11.2. I'm not sure what it is here. It doesn't like my points here. And I have 60 and 11.6. And I've still lost 20 and 8.6. So let's put that back in here. Not sure why it doesn't like my points here. So I'm missing 50 and 11.2. And 60 and 11.6. Frustrating. All right, so typically if these points were in a straight line kind of configuration, we would want to go in and do a best fit line. But notice that these are definitely not a straight line configuration. This is a curve. So the best thing to do here is just to trace the curve, connect the dots, something like that. Now what I want you to notice is that at first our line is really, really steep. And remember that slope equals rate. And then up here, our curve is actually flat. Up here, the slope is essentially 0. We'll talk about what that means here in a second. All right, let's go back and look at the rest of the question. So we graphed it. Calculate the rate for the reaction between 0 and 30 minutes. So remember, to calculate slope, to calculate rate, we want to calculate slope. Slope is equal to y2 minus y1 over x2 minus x1. Well, at time 0, which is our first point, it asks us to calculate the slope, first part of the interval, it's 0, 0. And at 30 minutes, which is the other point that it tells us to calculate at, is our point is 30 and 10.4. So to calculate the slope, take your y's and subtract your y's from each other. So that's going to be 10.4 minus 0. This y minus that y. Over, now we got to subtract our x's down on the bottom. Well, if we call this y2, this has to be x2. So 30 minus 0. This 30 minus this 0. So now we simplify that out. That's going to be 10.4 over 30. If we divide that out, we end up with a rate of 0.347, 0.347. And our units on that are going to be our y units, which are micromolarity, over our x units, which are minutes. So 0.347 micromolarity per minute. That's our rate. If you were doing this on an AP exam, you would want to show, you probably want to show this. You'd want to go ahead and show this. And then your answer with units. I could see them asking a rate question on this year's exam. Let's backtrack, and now let's look at part 2. Explain why a change in the reaction rate was observed after 30 minutes. So if we go back to our graph, up until 25 to 30 minutes, our rate is really, really, really high. That means we've got a fast reaction. But then after 30 minutes, again, our line slopes off to almost 0. The slope is 0. Well, if the slope is 0, that means the rate is 0. That essentially means that in this part of the graph, the reaction has essentially stopped. So that's what they're wanting you to explain. Why? So you would want to say that in after 30 minutes, essentially the reaction has nearly stopped. And the most logical explanation is that the starch, the substrate, has run out, or the concentration of the substrate is very, very low. Remember that enzymes don't get used up. So you can't say that the amylase is used up or that the amylase concentration is low. The starch is getting broken down and converted to maltose. So when there's less and less starch, the reaction rate is going to get lower and lower. That's the best answer for B. So C says draw and label another line on the graph to predict the results if the concentration of alpha amylase was doubled. Explain your predicted results. So we're doubling the enzyme concentration. Doesn't say anything about changing the substrate concentration. We're doubling the enzyme concentration. So what I would hope that you would understand, and let's see if I can erase a little bit of this, is if we double the enzyme concentration, that should initially double our rate. More enzyme is going to mean faster rate, which means we need steeper line, essentially twice as steep. So if the slope of this one is 0.347, we'd want to make a slope of our next one be about 0.7-ish. Maybe something like that. Now here's the tricky part of this. It still is going to even off because there's a set amount of starch. And once that starch is used up, the rate's going to go to 0. So you might want to say this is line 2 and this is line 1. And then in terms of explaining our predicted results, we would say that when we double the concentration of amylase, the reaction rate is going to essentially double. It's going to increase. Ultimately, though, the starch is all used up or nearly used up, and at that point, the rate drops to 0. We should get this essentially the same amount of product in this situation as we got in our initial situation, because the amount of starch and therefore the amount of maltose shouldn't change. All right, the final part of this question, identify 2 environmental factors that can change the rate of an enzyme-mediated reaction. 2 environmental factors. So there's lots of ways you could go with this. We had a long list just a minute ago that we talked about. The most obvious ways you could go here, I would maybe go with pH and temperature. And talk about if you move the pH in either direction, down and more acidic or up and more basic, away from the optimum pH for this enzyme, the rate is going to slow. Temperature. If you increase the temperature, as you increase it up to the optimum temperature, the rate is going to increase. If you continue to increase it past the optimum temperature, the rate's going to decrease. And in both cases, the discussion of how each of these factors affect the rate, talk about the changes. Higher or lower pH is going to slow the rate. Higher temperature up to the optimum is going to increase. Past the optimum is going to decrease. You might want to throw in that changing the pH, increasing the temperature above the optimum temperature is going to cause the enzyme to denature, change shape, and not function. All right, so that's it for our first FRQ. Before we move on, are there questions on this one? All right, we'll go ahead and do the second FRQ, and then we'll call it a day at this point, and we'll pick back up on bioenergetics next time. But here's the second FRQ. This one is really straightforward, and students, I really think that this AP exam may be the easiest one that we've had in years because they're trying to take a whole course and evaluate it with 2 questions. So I think the questions have to be very general. And this is a very general kind of question. So take a minute, read the question, think about how you would answer A and B. And this is really hard to read. This is identify. I think this is saying identify the likely metabolic process for step 1 and for step 2. And part B says identify an organism that carries out both processes. So here's what I'm hoping that you see. Notice for process 1, carbon dioxide is an input and an output is organic molecules. So carbon dioxide in, organic molecules out. Carbon dioxide as a reactant, organic molecules as a product. I'm hoping you guys see that that's photosynthesis. Process 1 is photosynthesis. It's taking in carbon dioxide and it's using that carbon dioxide and also probably some water to produce sugars, organic sugars. Those sugars then can be used to make other organic molecules. So we identified the process for step 1. That would be a point. You should say, complete sentence, process 1 is likely photosynthesis. And then the second part, explain how each process promotes movement of carbon through the cycle. For each process, your explanation should focus on the role of energy in the movement of carbon. So I might say something like, producers take carbon dioxide in from the air. They then use energy from the sun to combine these carbon dioxides, to bind them together, to form a sugar, an organic molecule. organic molecules as reactants in a carbon dioxide product. Well, I'm hoping that's cellular respiration. So I would say process 2 is likely cellular respiration. And then for the second part of that question, cellular respiration uses carbon-containing organic molecules like sugar as substrates. To go through as those molecules move through the process of cell respiration, energy is transferred from those organic molecules to energy carrier molecules like ATP and NADPH. And ultimately, the carbons in the sugars are released from the cell as carbon dioxide. Part B is really straightforward. You just have to know what kinds of organisms do both photosynthesis and cell respiration. And here's the thing. Every living thing on Earth does cell respiration. So really all you got to know is something that does photosynthesis. So you could say a plant, because plants have to do photosynthesis and cell respiration. You could name a specific kind of plant. You could say an algae, or you could name a specific kind of algae. You could say certain protist, or you could give a specific name for a protist, like Volvox, for example. You could even say certain bacteria, like cyanobacteria. So essentially, I would list one of those and say that plants are capable of doing both photosynthesis and cell respiration. Algae are capable of doing both photosynthesis and cell respiration. So this was a really straightforward, easy question, and I very well foresee something similar, just reformatted a bit, being on the AP exam. I'm going to go ahead and give you a preview of the next question we're going to look at next time. So this one is kind of an experimental setup. They love to ask these questions on the newer versions of the AP exam. So they're giving you some data, and then essentially they're asking you to explain that data. Oftentimes they'll ask you to make predictions based on the data and justify those predictions. This one doesn't quite go that far, but this question is looking at cell respiration. So next time we'll pick up with this question, which is labeled FRQ4 here. This is actually one of our checkpoint questions in our curricular materials, but for today, it's FRQ3. All right, before I stop, anybody have questions that I can answer for you?